Hello students. Now today we will discuss stem cells and various questions related to fertilization and embryogenesis in human. So let us start with that. So first of all, what are stem cells? Stem cells, we know these are cells, are the specialized cells which have a potential to produce any type of cell. It may be muscle cell, brain, nerve cells, clear, any type of cell can be produced from those stem cells. So that is called pluripotent. That means they can produce numerous type of, they have the potential to produce numerous type of cells. Now these stem cells are of two types. One is in human being, it is from embryonal stem cell, clear, which is taken from the embryo, and we know that embryo has the cells which produces various tissues and organs and finally the fetus. So embryo must have the stem cells, and adult stem cells are also there, clear, where the stem cells are taken from the, so you can see here adult stem cells, they are of two types. One is the taken from the adult tissue directly and used. So they can have limited potential. They can only produce the tissues wherefrom they have been collected. And another type is the adult tissues are taken and then their pluripotency is induced in them, and they have comparatively variety of cells can be produced by them, but very, because it is hard to produce this in inducing this pluripotency, it's very so less in use. Now embryonic stem cell, this is produced from the inner cell mass of the blastocyst, clear. Sometimes it is taken before the implantation and before the stage of it enters the implantation, and or it is taken just at the stage when it gets implanted, clear. So these inner cell mass of the blastocyst, these are the actually the stem cells which are in large use nowadays. And these stem cells, so these are the pluripotent stem cells because these are the embryonal stem cells, and you can see here these stem cells has the potential to produce more stem cells, as well as it has also the potential to produce specialized cells. So it produces both kinds of cells, that means more stem cells or the various types of specialized cells like blood cells, brain, neurons, muscle cells, etc. So various specialized cells are produced. In medical use, clear, in medicine, in medical purpose, nowadays the most used is these hematopoietic stem cells, clear, which is taken from the bone marrow, and this is used for producing various blood cells. So hematopoietic stem cells are the stem cells taken from the bone marrow and are used for producing various blood cells. But stem cells has the potential. So stem cells are nowadays used for the stem cell therapy is used for paralysis, treating the person with paralysis, heart diseases, clear, and it has also a potential to treat the person with diabetes, Alzheimer's. So this is about the stem cell. So most used is the embryonic stem cell. It is formed from the inner cell mass of the, or this is also, it's embryonic knob, because here from the fetus will develop. These are the cells where from the fetus will develop. So it has the potential to produce all types of cells. So this is the stem cell actually used, embryonic stem cells, and which has the potential to produce the small stem cell as well as the various specialized cells. Clear? And now students, we are going to discuss certain questions, clear, related to this fertilization and embryogenesis. So let us see. Often this figure is given. You can see here the figure. This is the ovum. This is the plasma membrane, then perivital space. This is the zona pellucida. These are the sperms entering. And the questions are generally they are asked you to identify these labels, clear, the ovum or the sperm or the zona pellucida. Name the state and function of this zona pellucida. Explain the role of hormones in the formation of that formation and release of ovum. That means the secondary oocyte. You have to tell the hormones, that is LH, FSH, and that estrogen and all. Draw the diagram uh, of B separately. That means here B is marked as, the sperm is marked as B. So you have to draw the diagram of the sperm separately and label its parts. Now there is another question you can see here. It is given, that same figure is given, and one of the sperms is observed to penetrate the ovum as shown in the diagram. How the sperm is able to do so? So you have to tell that first of all it will enter by penetrating the corona cells. It will enter into the, by acrosomal enzymes, it will penetrate the corona cells and it will enter the zona, enter and will touch the zona pellucida, where there are the, where from it will be decided whether the sperm will be able to enter or not, and various that this compatible reaction as the sperm touches the zona pellucida. The compatible reaction will occur because zona pellucida contains certain proteins which are responsible, and the sperm surface, the compatible reaction, if it is positive, then only it will enter, and as soon as it enters there will be cortical granules will be released and it will prevent any other sperm to enter into the, and the sperm should enter through animal pole also, that is also important. So this you have to tell. Next, difference between morula and blastula. What are the difference between morula and blastula? Morula, we know that is about 16 to 32 cell stage. Blastula is 32 above 32, that is 64 or like that stage. And morula, the cells are a solid mass, it forms a solid mass, but in blastula, you know that it forms the various, it is differentiated into trophoblast, inner cell mass, and zona pellucida is absent in morula, but zona pellucida is present in the, so you can see here, so it is a solid mass. Morula is a solid mass of 16 to 80 32 cell state, but blastula contains a fluid called blastocell, the outer covering called trophoblast, the inner cell mass which will grow to embryo. So this is the blastula. Zona pellucida has been broken. So these are the, so no there is a cavity fluid filled cavity, and this fluid filled cavity is absent in morula. Solid mass is there, no differentiation, but here it is differentiated into trophoblast and inner cell. Cells are differentiated into trophoblast and inner cell mass. Zona pellucida is present here, zona pellucida is absent here. Next question is, name the stage embryo gets implanted. Draw it and how it gets implanted. So what will be the stage? The blastocele or blastocyst or blastula is the stage when the embryo gets implanted. Clear? And you have to draw it. Just now we have seen the stage of blastula, that is inner cell mass, and this blastocele, trophoblast, all these you have to draw. And how it gets implanted. So what happens when it moves to the this region, when it moves to this uterus? Clear? When this embryo moves to this uterus region, this uterine fluid is released, the blastula enlarges in size. Clear? And then the trophoblast differentiates into trophoectoderm and trophoendoderm. Trophoectoderm forms the chorion. Clear? The trophoectoderm forms the chorion, and this chorion produces the chorionic villi, and this is the embryonal wall. This chorionic villi enters into the embryonal wall, breaking the embryonal, and this embryonal wall and the chorionic villi gets interdigitated, and thus it gets attached with the endometrial wall. this chorion produces chorionic villi, and this chorionic villi enters into the endometrial wall. This is the endometrial wall, and this gets this endometrial wall and the chorionic villi gets interdigitated, clear, and forming the placenta, and thus it gets attached with the endometrial wall. So this is called implantation. So just so first of all, blastula enters into to the endometrial wall. The uterine milk is produced. It is enlarges in size. It gets embedded with the endometrial tissue. The trophoblast differentiates into chorion and amnion. This chorion produces chorionic villi. Chorionic villi moves into the endometrial wall, gets interdigitated, forming placenta, and thus the whole embryo gets attached with the endometrial wall, and this is called implantation. Next states the fate of trophoblast and inner cell mass. This inner cell mass will develop to first of all it will produce the germinal layer, and later on it will produce the fetus. Clear? So inner cell mass, first of all it will produce the germinal layer, and later on it will produce the fetus. And this trophoblast will differentiate into trophoectoderm and trophoendoderm, which will produce chorion and amnion. Okay, two layers of the encircling the embryo. When and where chorionic villi appears, clear, and state its function. So already we have discussed, it appears at the blastocyst stage in the uterus, clear, and its function is to produce the placenta getting interdigitated with the endometrial wall. Structure of placenta we have discussed. Function of placenta, we know it is the placenta is the region where from nutrition is provided. The it provides antibody, respiratory gases, wastes or carbon dioxides are collected, germs are prevented to enter, and it also acts as endocrine gland. Now, how placenta acts as an endocrine gland? Justify. So we know that placenta acts as endocrine gland because endocrine glands are the glands which secretes hormone, and placenta also secretes lots of hormones. We know what are they? Certain hormones are released only by placenta only during pregnancy, like human chorionic gonadotrophin, human placental lactogen, clear, and relaxin is produced both by the corpus luteum and the placenta. And placenta also produce certain hormones which are produced in normally also, that is progesterone, estrogen, etc. So placenta produce lots of hormones, so it acts as an endocrine gland. Next is, what is pregnancy hormone and why it is so called, and state two sources. So pregnancy hormone, we know it is the progesterone, because progesterone actually maintains all the development related to pregnancy, and two sources of progesterone, one from the ovary, that is corpus luteum produces progesterone, and other from the placenta. So two sources of progesterone will be corpus luteum and placenta. Name the hormones secreted only during pregnancy, what are its source and function. So hormones secreted only during pregnancy will be, first of all, HCG. It is secreted from placenta, that human chorionic gonadotropin. It maintains this corpus luteum for the secretion of progesterone. HPL, human placental lactogen, which is also produced during from the placenta, and it helps in development of breast during pregnancy. Relaxin, it is another hormone which is secreted by placenta as well as corpus luteum. It is secreted during the parturition and helps in the softening of the pubic symphysis to make the parturition easier. What is parturition and why it is called neuroendocrine mechanism? Parturition is the childbirth, we know. It is called neuroendocrine mechanism because actually where from the signal originates? The signal for parturition originates from the fully developed fetus and the placenta. So signal for parturition originates from fully developed fetus and placenta. Both are the region where from the signal of parturition originates, and this is so this signal is neural part, and as this signal start originates, there is the release of the hormones like oxytocin from the posterior pituitary. There is increased receptors of oxytocin in the uterine muscles, and the posterior pituitary starts releasing hormone oxytocin, and this causes vigorous contraction in the uterine muscles, and where helps the fetus to move, get released, clear. And placenta also produces, along with the corpus luteum, produces relaxin during the childbirth, that also helps in softening the pubic symphysis. How milk production is regulated during pregnancy. How milk production is regulated during pregnancy. We know that prolactin is the hormone which helps in production of milk, and oxytocin helps in release of the milk when the child starts sucking. So prolactin produces the milk, helps in production of the milk, and oxytocin helps in release of the milk when the child starts sucking. And what is colostrum and why it is so significant? So we know that colostrum is the first milk after the childbirth, which is pale yellow in color, and it is very significant because it contains lots of nutrients, lots of antibodies like IgA, IgG, IgM. Clear, it contains growth factors, it contains laxative, so it is very much useful for the newly born baby for the next, to move to the next phase. So I think we have discussed more or less all the questions which can be coming from this part. So it's okay. Thank you all.